Well, we're here, so why don't we get underway?
You have to be on the mark.
I think everybody has met everyone except Eugene Schneider, who has been with us several times in the past, but Eugene, welcome.
Thank you.
Eugene serves on our DSMB, has served on STAR and a number of committees, and is the CMO at Ionis, and so brings a great deal of drug development experience and experience with antisense. So what we thought we would do in the next few minutes is just flesh out a little bit some of the challenges of managing these complex risk-benefit judgments that we have to make. And of course, you've heard a lot about that from Julie and from Lori and Sarah and others already. But I think the way I want to do this is to begin at the beginning with the phenotype. And maybe the most interesting example of a really difficult decision we had to make was with alternating hemiplegia of childhood. This is a disease that we can certainly help. We had every reason to believe that we had an ASO, that we could make an ASO that would work. And ultimately, we decided not to pursue that patient. And I think it was mentioned earlier in the day that the reason we chose not to pursue it was that an in-depth examination of mutations and phenotype associated with that gene led to the conclusion that there was a possibility that reduction of that target at some point in the life of that patient could result in an unpredictable, extremely rapid, very rapidly fatal form of Parkinson's disease. And that was the subject of intense conversations and they were complex conversations, because do we not help this patient because that could happen in the future? Is the risk really that great? Is there a way that we could identify that risk and predict when it might happen and whether we could stop the drug in time? These were all things that we had to talk about. So maybe I'll turn to Eugene and talk about the deliberations about that patient and how that went.
Well, it was probably mostly at the Access to Treatment Committee, I imagine, which tackles these types of tough questions and try to understand molecular biology and the phenotype and how, if it's appropriate to thread the needle for a target to engage the target that has potentially some liability of over-engaging. That's exactly what that committee does. They look at science, they look at what's available, and try to decide whether it makes sense to pursue that further and continue the investments. I haven't been on that committee, so I don't know the particulars of that particular discussion, but it seems that that's what we would tackle exactly.
Yeah, and of course, Lori, you participated extensively in that discussion, as did I, and our CMO, who's not here today, Joe Gleason, was a principal component in that conversation. Want to talk about that?
Yeah, so I mean, just like Eugene said, that's really the committee where we will discuss all that. And there's a lot of diseases, or disease that we look at that have a spectrum, right? And so you have a spectrum. If it's a gain of function mutation, causes syndrome X, it can be very severe. But if you knock everything down, you cause another disease, right? And again, the last thing we want to do is add burden to patients, so we can't go there. And what we had realized here is even if we did an allele selective ASO, we would still potentially induce this other disease that was really bad. And the problem was that you couldn't determine when this could happen. Because this also could happen if the patient has an episode, of some infection or something. So we didn't know if it could happen, when it could happen, but we knew it could happen. And just the idea of potentially inducing something in a patient that is irreversible. And that was the problem. It was also an irreversible phenotype. So we couldn't even say, sometimes if you induce too many seizures because maybe you knocked it down too much, you could stop the ASO and then you can go back. But in this case, we couldn't do that, even if you stopped the drug. And so it was a hard decision. This was a 4-year-old little girl who really needed help. She had hemiplegic attacks a lot. And, but we decided that unfortunately, this was just not
It was a conversation that took months. I personally had to really dig into the literature to convince myself that the risk was palpable. Ultimately, it came down to, the opinion was still divided. And ultimately, I had to make the decision. I decided not to treat that patient. Those are incredibly hard. To know that we could potentially help a child and walk away from that opportunity because we could potentially do real harm to that child at a later point, and not have any information to tell us how big that risk was. It was, as you would expect, very contentious and very difficult. And the reason I pick it first is that it happened in the very first step in considering whether this patient, who was treatable from an ASO perspective, had a syndrome that needed to be treated, could be treated long-term with sufficient safety to warrant going forward. And it was a decision that we came to that was, no, we should not do it. And so that's at least something that I think would be easy to have overlooked. Without an in-depth understanding of the phenotype, the disease, all of the literature work that went on behind the scenes, one could easily just jump in and do something that could do harm. I don't know. I am comfortable with the decision. You're comfortable too, Lori?
Yeah, in the end, we were, we still wish we could have helped this child, but.
And as of course, we're continuing to follow that literature, and as we learn more, if it turns out that there's a predictive test or something else that we could do, we'll reverse our decision and go forward, because we know we could help that patient and we could help other patients like it. And then I think Julie mentioned a couple of places where we run into issues. And she talked about tox, but our success rate in our GLP tox studies is what? About
90%.
About 90%. So those examples in tox are actually rare.
Yes.
But they're rare because of the work we do before
Upstream,
we get there.
Yes.
So what would you say our success rate in tolerability studies is?
In terms of ASO, or in terms of number of ASOs or number of programs?
Number of programs.
It's probably in the same range, I would think.
Mm-hmm. So we will lose ASOs throughout. If the BJAB assay is worrisome to us, we may discontinue 15 of the 20 ASOs we have or more. Mm-hmm. If the off-target analysis causes us concern, we'll discard some ASOs there. And so if you look at our cumulative, if you look at the success just in tox, it looks really great, which it should, because of all the work that we've done that have weeded out all the potential problem ASOs before we get there. And we do that because the tox study is the bulk of our cost. And it is also the thing that consumes 6 months. Mm-hmm. So if we make a mistake in tox and we lose a drug in tox, that means we've spent a year to get there, 6 months in the toxicology study, several $100,000 plus manufacturing, plus sterile fills and everything else. And that means dollars that we can't spend on the next patient. And so we try to minimize that. So I would say it's that cumulative weaning process that is the critical thing that leads to the 90% success rate.
And that's why we want to start with the highest number of ASOs as possible in the beginning so that we have room to narrow it down as well.
Yeah. So we start, let's say we start with 400 or 500 and we end with 1.
Yes. Sometimes we have a backup.
Yeah. And we always try to have a backup.
Yeah.
But any place in there, Julie, that you particularly worry about, that where we wish we knew more, that we could cut down the number of ASOs we lose?
I think I would say a year ago we were losing more ASOs in tolerability, and then we, with the help of our friend at Ionis, changed the formulation of the ASOs, and that helped reduce also the immediate reactions to the ASOs because of the salt content. So that made a real difference.
Yeah, so that's a really interesting point. When we administer an ASO intravitreally, intrathecally to a rat, the rat has about 300 microliters of CSF. That's a little tiny little bit. That's very little. And when we administer an ASO in high concentrations, we can actually chelate all the divalent cations out of the CSF. And that can lead to very odd behaviors in that rat because you need magnesium to do what you need to do in your central nervous system. That took us years and years to learn. And actually, some time ago, I don't know how long ago, Scott, we began to put magnesium in the...
I wish we'd started earlier.
Well, you will recall, I said this is chelation 20 years ago.
Well, we started out with some supplemented cations, but in our tox studies, we could have started increasing that supplementation a little earlier.
Yeah. So we lost a lot of ASOs at Ionis that we should have lost because we didn't get to the bottom of this problem as quickly as we might have. And that means, I guess the point I want to make there is that you pay attention to the data. And you learn from looking at the data and cumulative information that only comes with millions of ASO experience, and then you apply that and solve problems that cost you good quality ASOs in the past. And so we don't lose ASOs that way anymore.
Right.
And both at Ionis and at n-Lorem. And at n-Lorem it matters much more because our patients are in immediate need and every dollar and every day matters. So most of our patients are CNS patients, and so the things that we worry about in the central nervous system are two things, really, now that we've sorted out cations and supplementing with magnesium and that sort of thing. And they are really cytotoxicity, which can be due, we think we understand the mechanism there, and innate immune activation, which is very complex. You want to talk about Purkinje cells a little bit, Scott?
Yeah, so that's a particular cell type in the brain And it really is sort of notorious for being one of the more sensitive neurons to any kind of environmental or chemical exposure that could be harmful. And so one of the things that we've seen is sort of our sentinel in terms of screening potential compounds and weeding out the bad actors, as Julie was saying, was to keep a particular eye on this the cell type in the brain. I think that's one of the things that's come from experience, and over the course of multiple programs, we know precisely now the very specific sort of endpoints we need to look at as we're sort of identifying that perfect compound that's got good potency and good tolerability. And so neuronal degeneration of any kind is absolutely the sort of thing that we want to avoid. We know what the sentinel cell type is. We know the mechanism generally that causes harmful things to happen in Purkinje cells, and so that even goes into the design and the bioinformatics stage of sort of designing oligocandidates and decreasing the probability of that. So through mechanistic work, through the careful sort of observational work, that's one of our main sentinels to make sure that we're picking compounds that are not going to have any particular sort of neuronal toxicity, or at least reduce that probability as much as we can.
So if you're asking, what do we look at first? Well, typically, what I want to know first is, are there any Purkinje cell problems? And if we see Purkinje cell problems, that means we have an ASO that we should not move forward. And so if we see that, it's over. And fortunately, we don't see that very often because of all the pre-screens that we do. We've seen a couple, I guess.
Yeah.
Yep, they occur.
Yeah.
And that's part of the, what we lose in the screening. And again, the endpoints we have are very precise. We can do specific staining for those cells, so it's very easy to pick up a compound that's going to have a problem.
Yep, and specifically this problem is what caused us to discontinue the tardy BP case that I discussed. It was Purkinje cells.
Yeah. You remember Julie talked about tardy P, and we had to walk away from the ASO that we originally picked. It was because of Purkinje cell death. And we saw enough problems. If we see even a hint of Purkinje cell problems, that's just too risky for us to move that ASO forward. So once again, would you know this if you hadn't spent 35, 36 years at this? No, you wouldn't. You might say, as we did when we first saw it many, many years ago, well, who cares? It's just the Purkinje cells. We learned the hard way that the Purkinje cell were trying to tell us something, what they were telling us was important that we should listen to it.
Yeah, and I think the spirit and the culture you established at Ionis, and now carrying over to n-Lorem is to put that information out there. So our issues with Purkinje cells and our mechanism and our understanding of those are, even from my Ionis perspective, is sort of out there in the scientific literature. So it's informing the broader community.
But you have to read it.
And that's true. Yeah, it's out there, but
I mean, we've published this now so many times, and yet it's astonishing to me. There are people making ASOs in the world who haven't read any of that literature.
Yep.
And if you haven't read it, you can't know it, and if you can't know it, you can't be warned by it, and that's the value of experience. I think maybe the next thing that would be worth talking about would be fever. I think the fever associated with dosing in our KIF1A patient, Susanna, consumed all of our time and best efforts for many, many months. I don't know, Laura, you want to just talk about how it came about and.
Yeah, I mean, I can start, and I'm sure everybody here can add to the story, but I mean, basically, like I said, we track safety over time, right? And so we put data packages together for the DSMB to review, and so we always have this slide where we look at when the doses happen, when the adverse events happen in relation to the doses, and so after a while, we saw that there was always fevers for Susanna after the dose, and we went back, and I went back to talk, to Jen about it, and I said, what are those fevers? How are they? What's going on? And we realized, they were getting longer or stronger at the beginning, and so then Jen decided to pre-treat with steroids to help. But then, we discussed it internally, and we're like, well, is this normal? Like, should this happen, or should we just try to better understand what's going on? And even at the DSMB, we had like, so, what are we doing? And so then we all came together internally and with Jen to say, okay, we really want to do some more analysis to try and understand this. And we have CSF samples that we collect every time before we give a dose. We pull out 20 mils of CSF and we push in 20 mils of drug. So we had that, right, and they aliquoted. And so we said, well, we have that, we need to analyze it, but we need to do this in a very systematic fashion and, with the right labs that know what they're doing. But also, we want to make sure we have enough CSF left in case we have to do follow-up analyses. And so it was really going through all of that and for us trying to find the right lab to do this. And this kind of goes back then to all the experience that Ionis had with that specific lab that ran all the innate immunity and complement work, for the systemic side of ASOs. And so we were able to really work with them and kind of together really look at the data and, start analyzing more samples from the beginning. And we also analyzed samples from Sloan because we didn't have a baseline and we don't know what the pediatric values are. So we needed to get another pediatric patient.
Yeah, it was even more complicated than that, if I remember correctly, because one of the anesthetic that was being used is associated with fever, and the disease is associated with fever. And so the first thing I think we did was change anesthetics. That didn't help. And what we were really worried about was inflammation. And inflammation in the central nervous system, we believe, most of the time reflects activation of the most primitive of your immune system, the innate immune system. And we've learned a lot about innate immunity and how it gets turned on, and we have a paper that we hope to resubmit soon that shows how it gets turned off, which is kind of cool. And so what we were really worried about there was, is there the possibility that we have an inflammatory process that could increase the amount of CSF and increase pressure in the brain, which can be rapidly fatal. Or if it builds slowly, it can compress the brain, and that presents as normal pressure hydrocephalus. High pressure hydrocephalus, you can see the pressure go up when you measure it, when you do a spinal tap. But if it builds slowly, the brain compresses and you have normal pressure. Either way you do it, you've had too much CSF, too much pressure in the brain, and you're doing harm. And so this was really a deep concern. And we had the experience to understand the impact of an inflammatory background. All of our patients have chronic disease, so we would expect all of our patients to have some inflammation of chronic disease. And that just increases the risk that you can get inflammatory responses in general. And so we had all that experience from Ionis, plus we had experience in non-human primates where we can, because they're complement system is slightly different, we can see activation of complement, and complement activation is a very dangerous thing. Now, you need it when you need it, but you don't want it when you don't got it. And what we were really worried about was, are we activating complement in the CSF? That question really has not been asked in a real way. People have looked at Alzheimer's and Parkinson's at complement levels, but there are no normal values in the CSF. There's no normal value in a child. So the question was, how do we figure this out? Thanks to our previous experience, we knew where to go.
Yep.
National Jewish, I guess, these are guys you worked with for a long time.
Yeah, I mean, it was the likely suspects were one of these two pathways, either sort of cytokine production or altering the complement activation. So, and again, that's where the non-clinical work which I do, and translating that to human is a tricky thing sometimes. And this was a curveball we didn't expect, because we don't see fevers in the animal studies when we're doing the safety testing and stuff. But getting the team together to sort of work through the issues and again, kind of using our experience that we'd established over the years, with both local and systemic administration about what pathways to look at. I think, even though it was a very complex, chaotic, situation at the time, put us on the path to, a couple of investigative follow-ups that I think, we can help to use to sort of manage the patient at this point, try to help understand things better.
Now, the first call I made was to Eugene to see, because I didn't remember ever seeing a fever after dose after any intrathecal administration of any ASO that we'd ever done. And I don't think we still have, right, other than with Susanna.
No, we see a lot of fever in pediatrics in general. Obviously, this is where, again, you see this clear difference, as they say, strength in numbers. When you run typical clinical trials and you have groups of patients, you can observe groups, you can observe sort of frequencies of those with an adverse effect versus those without. You could make some inferences to whether it's potentially related, what are the patterns, what are the similarities. In this situation where we have one patient, we have to really very painstakingly analyze all of the information that is available. It becomes a real challenge to, sort of rely on that experience from large studies to try to figure out what's happening. Not only that, but again, oftentimes in trials, obviously investigators are free to discontinue patients, and oftentimes that's the first reflex because, again, there are groups of patients, and once the groups have been tested, that's when you learn about sort of patterns and severity, and you don't have that luxury, so that decision is essentially irreversible to discontinue treatment. That means the drug is dead, and there's especially it's often kind of what's staring us in the face is the benefit side, as Lori said. This is I think of it as a balancing beam, and you really want to make sure that the benefit is really far exceeds whatever the risk or potential inconvenience of drug treatment is. So that decision, that irreversible decision to start treatment and to expose the patient to essentially the lifetime of risk from disease progression is a really important decision. So we really need to be careful, and that's where that art piece comes in, and you try to piece together the evidence.
Yeah. So we called in a bunch of favors. We got a whole bunch of assays done that nobody does on CSF. And we know from our work that the reason some ASOs activate complement in the monkey is that a certain part of the complement system, factor H, which keeps complement from activating, is different in monkey. And so we looked at then factor H and asked about complement activation, because we saw a lot of complement activation that was likely ASO caused. It would just be too risky. We evaluated that. We did not, the pattern was odd, but certainly that did not implicate a mechanism that we know to be an ASO mechanism. And so we arrived at a path to continue treating, but to watch CSF pressure carefully, and to continue to watch the cellularity of her CSF and other things to make sure that we don't cause a significant problem. That was, again, months of effort in multiple labs around the US, and many, many conversations with Jen and the entire team. And that happens. These things are complex.
I think the other element of the experience that I thought helped manage this is, again, just kind of our broader knowledge of what the dose response is from bigger trials, and, having the confidence that, we could, or you could adjust the intensity of dosing, but still, remain in a range, that's likely to still be, providing benefit. A lot of PK, a lot of exposure, dose response experience that we can draw on.
Yeah, and our own experience. I mean, 6 years ago, if somebody had asked me, would 20 milligrams intrathecal actually produce a positive effect, I'd say no. I would have been wrong, I'm happy to tell you. We see all kinds of positives at 20, 40, 50, 60. And so armed with all that, we knew that we could lower the dose, still see benefit, and we could still take care of her and treat her safely by just checking a few extra boxes every time we treated her. And so that's, again, the sort of challenge that presents itself in real life situations where we're taking advantage of as broad a universe of knowledge as possible really plays an important role. And recognizing that we're all fallible, and that above all, we don't want to hurt. And bringing the group together, doing the necessary work. We're continuing to do work in this area. We're continuing to watch cytokines. We'd like to develop normal levels of all these analytes. They don't exist. So we don't have any yardstick to help us. And as we gain more experience, we'll create more of those yardsticks that we can use. The next time we run into fever, we have this experience to help us, and that will be important. Yeah.
Yep.
So what have I missed, guys?
No, I mean, and I think it gives us the opportunity, what we learned there. I mean, we're treating a lot of kids, right, with disease, so we will never be able to collect enough CSF from normal children to be able to run all these assays, but we have all this CSF at our hands, and we know now what maybe we can use to create those normal values. And they're not necessarily normal because the patients all have underlying disease, but at least we could get ranges. And so I think it's nice from a research, scientific, curious mind perspective, like we now have something else that we can go after and try to analyze potentially across the board of all our patients that we're treating.
Yeah, and we can take our other KIF1A patients and collect CSF cytokines and complement and begin to create a database that'll help us the next time we run into a fever.
Yep, for sure.
Yeah, I think one other comment I'd make in credit to the way that this complex case has been handled in terms of just the interaction with the FDA. And I really think it's hard for the audience to appreciate how remarkable the guidance is that came out in terms of both the timing as well as just sort of the flexibility that they broke all of their standard paradigms, and as I think was reviewed earlier, tolerability has been good by and large. The speed bumps that have come up, it's incredibly important. I give great credit to the fact that that's been communicated back to the FDA, to sort of reciprocate that trust, or solidify that trust that they basically had in the community to sort of put those guidance out there in the first place.
I think the guidance exists because of the properties of ASOs and because of the behavior of Ionis over 30 years. And we've tried to continue that same commitment to responsible behavior at n-Lorem, and I think we're doing it.
Yep. Yeah, definitely worth a specific mention of that, I think, so.
Yeah. Julie, anything you want to add or subtract or?
No, I think back to this point, the ASO is the perfect modality for the nano-rare patient because it gives you the opportunity to stop treatment if something wrong happened, compared to gene therapy where the treatment is a bit more permanent. So I think, perhaps it explains also the flexibility of the FDA, but the guidelines that they've created are working because we're seeing safety in all of our trials, and I think that's really important as well.
Yeah. Maybe we talk about cure for a minute. Cure is a word that is used a lot. And cure implies permanent. And in truth, drugs don't cure. The only drugs that cure are anti-infectives because it's an infectious disease, it's an external organism. And then today we cure some cancers. Beyond that, we don't cure diseases. We prevent, treat, so on. And while it would be great to think about a single treatment curing, the issue with the single treatment curing is that that single treatment produced a permanent change. And that would be great if the permanent change is all good. But what happens if that permanent change is not good? And we see that in a variety of places where cure is the goal. And so what Julie had to say, which is that ASOs eventually are cleared. And they steadily reduce in concentration. So even if we produce a side effect, if we protect that patient and care for that patient through that side effect, it will go away. That's our job. You can't do that if you have an agent that you think is going to produce a permanent change in that patient. And so maybe someday a cure would be a good thing to have. Right now, if it were my child, I would much prefer, given what's known and what's not known, to have a drug that treats that will go away in case there's a problem. Just food for thought for folks. Those options will come. And they're much earlier in experience and development, and we're excited about things like CRISPR and other things, but there's a lot to be learned before instituting a permanent change in a human being. Yep. Anything else, folks?
Well, since you mentioned prevention, what are your thoughts? May I ask you what your thoughts are, Stan, around prevention? And we kick it around quite a bit these days, especially now that genetic testing is more accessible, more prevalent. There are lots of questions related to sort of penetrance and when to start treating. But any thoughts on whether you see the future use of ASOs and related platforms as preventative, just like we use vaccines?
Absolutely, that's the future of medicine. I think the future of medicine that we're actually practicing the future of medicine every day at n-Lorem. That, to me, is in some ways the most exciting thing about what we're doing. We are we are identifying the cause of problems, treating the cause with a molecular treatment designed to address the cause, not the symptoms. We don't even ask what the name of the disease is. 9 times out of 10, I forget. What's DRPLA? I don't know what it means. But I know what the cause is, and I know how to fix it. That's what matters. And I think if we couple what we can do today with sequencing of newborns, all newborns, to identify those patients at risk, then we will have to deal with the issue of when to begin treating. And you know, there's a growing experience with that, with Huntington's, with many of the triplet repeat diseases that have a long anticipation period and so on. And there's experience that we have had at Ionis with things like SMA, where when we began treating patients with spinal muscular atrophy, we had to treat type 1 babies who, before treatment, the average time to death was 6 months. If you can imagine having a baby and losing it 6 months later. And we had to begin with treating those patients, and it was terrifying. That was the scaredest I've ever been doing a clinical trial, the first baby we treated. And as we learned more, we could then treat the less severe patients. And then we did the nurture study with Biogen, where we treated pre-symptomatic patients. And I'm still in touch with some of those folks, and they're 9, 10, 11 years old, and they look healthy. Other than getting their injections every little bit, they're healthy. So I think the answer in the future will be sequence all newborns, identify patients at risk, have genetic treatments that address the cause of those problems, and be ready to treat at the earliest appropriate time. And each disease will be different. You'll have to make a different decision for each disease, but that'll be a high quality problem. I mean, think about it. Wouldn't that be a problem you'd rather have than trying to take care of your patient when they've progressed so far that you don't know if they'll live long enough to get treated? We have bad days. We have days when somebody we hope to treat doesn't have the time. That should never happen. And in the future, it won't happen. That's the future I want to drive.
Yeah. Yep.
Sound like a future that you want to work toward?
Absolutely.
Hey, I'm a dreamer. It's what I do. Maybe we do questions, or? Yeah, we got time.
Yeah, 7 minutes.
Oh, no questions.
Hello, Nathan Guo, ZTTK syndrome. I guess given some of what you've described about all the different data points that are important to gather, but clearly an evolving set of what are the right features to gather, if you were a parent thinking about how can we make sure that we're gathering the right data from our community, et cetera, what would be the main things that are most critical to gather? Obviously hard to generalize across diseases, but from a neurodevelopmental perspective, from a clinical data perspective, what are the things that parents should be thinking about? Thank you.
I mean, I can start. I'm sure everybody can talk here. I mean, every data point would be important, but I think even when we think of the fevers or anything like that, right? So reactions to any anesthesia, for us to better understand how did the child respond to an anesthesia for some whatever procedure before, right, to after, if that's part of the, it's not part of the medical history, but if that could be like the write-up in some medical, clinical notes that we get. I think those types of things are always important, but they're never really necessarily collected anywhere. But then if as a community, you can do studies in a community to try to assess those things, do people get a lot of fevers? I mean, that's why we find out about the fevers for T408, right? But things like that even show us like the safety, in a patient population or in a patient. I think for me, that would be something to collect that is normally not collected or not easily collected.
Yeah, I would definitely add to that that you have a much longer observation of the child than even the most experienced investigator or site personnel do, right? You know your child best, and really trying to convey that color, that context that is going to be important to interpret both the goods, right, the improvements, the stabilization, as well as potentially side effects, is going to be important because it's all coming back to judgment. What does this mean? And how does it relate to my child? And no one can answer that question in a vacuum, no matter how experienced those investigators are. So it really is being able to provide that valuable context that you live day in and day out. Interpret the fevers, interpret the little things that seem trivial but may not be, because these are important pieces of data. We're trying to aggregate everything into one sort of cohesive story and try to observe it over time, and it's difficult.
One of the many exciting things for me is that we're really becoming much better at using caregiver information. That is information that has been discarded for now the better part of a century. As, you know, too biased, too this, too that. All of that's true, but learning how to use that information, sort the bias out, and combine that with empirical, measurable data, I think is going to be an important contribution that n-Lorem makes to medical science, and that's really, I think, vital. You know, the thing that I wish we had more of would be natural history information, but every mutation is different.
Yeah.
And so getting natural history information for a disease, which used to be satisfactory, is a waste of time for me. I mean, it helps, but what I really need to know is, for that particular type of mutation, what is the natural history? And so we sort of work in the dark, and I expect to keep working in the dark for quite a while, because we're at the edge of sophistication in what we're doing, and people thinking about diseases, and I'm thinking about mutations, and I know that even with the same mutation, you can get a different phenotype, and with different mutations, you can get widely divergent phenotypes. And so what we'd love to see is natural history studies that focused on the cause and compare different types of mutations and their natural history within that disease category. And I'm afraid that's a long time coming.
I think, Melody, we'll take your last question, and then I have a bunch that are coming in online, and I just want people to know that we'll try to get those in today, but just to keep us on time, Melody, I'll just let you ask the last question.
Thanks, Amy. Hi, everyone. I'm Melody Kaiser. I'm here with Families SCN2A Foundation and also the Global Leukodystrophy Initiative. I have a question. When you're doing your risk-benefit analysis about whether to move forward with a program, are you engaging with patient advocates and bioethicists? Are they part of the conversation?
I wasn't able to hear the question.
When we do our benefit risk analysis during the entire process, right? You mean even the ASO design and discovery?
It's throughout the whole process,
Correct.
but specifically when you're making that decision of, you know, you don't like the risk of, you know, inducing a loss of function or gain of function problem with your ASO and you're not sure you can do it safely.
Right, yeah, so do we engage patient advocacy groups and families at that time? I would say I'm not sure, not during the ASO, and anybody, Julie, you can also correct me, I wouldn't say during the ASO discovery process, we wouldn't at that point necessarily bring in patient advocacy. I mean, we would talk to physicians and patient groups more about the phenotype maybe, or the symptoms that we want to treat. Less, I think, the benefit risk ratio when we talk about a certain molecule to move it forward. I mean, if that, and maybe I didn't completely understand or answer your question, but I don't think we do. It's really, the patient advocacy group really comes at the point of treatment goals and trying to see the right outcome measures to use and things like that. And then if we present it, in two families about an adverse event or something like that, then, obviously we're happy to hear from even the advocacy groups and the families to see what they would be comfortable with in terms of, going forward with this or not, depending on the risk.
Thank you. I know, speaking from the advocacy groups, a lot of us would love a voice at the table to help inform some of those decisions. So I'd love to chat with you more about that. Thank you.
Appreciate that.
All right, thank you for our panel of experts here.
I don't know why I'm going off. I'm sorry.